This unit is organized by big idea. So we're going to look at concepts related to ecology and relate it to some of these big ideas. Big idea 2 states that biological systems utilize free energy. So we're going to talk about how organisms like autotrophs acquire free energy. We're going to talk about how organisms get molecular building blocks like the carbon, hydrogen, nitrogen, phosphorus, sulfur. So we're going to look at those cycles so that they can build their organic molecules so that they can grow. We're going to take a look at plant growth and how it occurs at a cellular level. And we're going to look at reproduction, talk about things like diapause, and also about just maintaining homeostasis. We talked about organisms have to maintain certain levels, like certain temperatures and so on. Free energy must be supplied for a system to be maintained. If you think about your room is not going to clean itself or stay clean by its own. You're going to have to supply free energy to maintain that system or to offset entropy. A good example of a process that offsets entropy and uses free energy to do so is photosynthesis. In photosynthesis, plants are able to take in inorganic carbon as CO2, water molecules, and they're going to use light as the free energy source to make their glucose, C6H12O6, and other organic molecules. And then they make oxygen as a byproduct. When you look at the reactants of photosynthesis, you can see that they are more disordered. This is showing high entropy. The glucose molecule that is made is showing less entropy. It's more organized. And it took light energy or free energy to organize those molecules into a glucose molecule. Homeostasis means a constant, non-changing internal environment. Organisms have to maintain things like water and solute concentration. Plants and animals have to monitor the amount of light they're receiving. And we've talked about organisms sometimes have to maintain a certain body temperature. So we'll look at temperature regulation. In the past, we already looked at 2 thermoregulation strategies, 2 ways that organisms use energy to maintain homeostasis, to maintain body temperature. Ectotherms, we said, uses external energy sources to help regulate body temperature. Examples include fish, amphibians, like your frogs, and your reptiles. Whereas endotherms use internal energy sources to regulate body temperature. They're relying on reactions releasing heat to increase body temperature. I'm not going to spend a lot of time on this because we already talked about it, but you should recognize that as environmental temperature changes, you can tell this is the line of the ectotherm. Because if it's colder, let's say it's 10 degrees Celsius outside, then the body temperature of an ectotherm is going to be 10 degrees Celsius. So you can see temperature is changing as the environmental temperature is changing as well. Remember, some endotherms include birds and mammals. Also note that your endotherms are going to need more of a constant supply of free energy because they're going to use a lot of energy just to maintain body temperatures. You have to feed a mouse more than you have to feed a snake. Again, I don't want to spend a lot of time on this because we've already covered this before, but it is talking about free energy relating to organisms in an ecosystem. Remember that your endotherms, when they are in a colder temperature, they're going to have a higher respiration rate. You can see that this one is higher than down here. This one's lower because this is in a warmer temperature. Organisms that are endotherms are going to shiver in a colder environment, which is going to generate more heat. And to shiver, it takes ATP. So that's why these organisms are taking in more oxygen. It's because they need to make more ATP so that they continue to flex their muscles and generate heat to maintain body temperature. Whereas your exotherms, in a colder temperature, you can see that respiration rate decreases because they're not able to move as quickly. They're not able to breathe as quickly. Their heart rate drops because their body temperature is lower and all the reactions are taking place at a lower speed. When they warm up, though, is whenever you're going to see higher reaction rates and they're able then to take in more oxygen to make ATP. So again, mice are going to have a higher respiration rate in colder temperatures, whereas your crickets, which are your ectotherms or your exotherms, they're going to have a lower rate of respiration whenever it gets colder. Another idea to remember is that body size matters. I have two endotherms. I have a mouse and I have a rat. And you can see that oxygen consumption is changing due to colder versus warmer temperatures, which we talked about above. But also notice that the mouse is taking in more oxygen in a period of time than the rat. Since the mice are smaller, they're going to have a higher metabolic rate and a higher respiration rate. They have to bring in more food per gram of tissue than the rat because the rat is bigger. If you think about it, on a cold night, what type of organism is more likely to freeze to death? A mouse versus an elephant? Well, obviously it's going to be a mouse. A mouse has a hard time maintaining body temperature because it has a lot of surface area to let off heat, but not a lot of volume to generate. Whereas an elephant has a lot of volume, it can generate a lot of heat, but it doesn't have a lot of surface area to release the heat. So again, there's a relationship between metabolic rate and body size. Generally, the smaller the organism, the higher the rate of respiration. If you remember the video on the shrew, if the shrew misses a single meal, then it can die. Again, here's some questions related to ideas related to respiration and temperature and body size and so on. The correct answers are marked there for you, so you can take a second to look at those on your own. We've been focusing on animals and using the environment or using internal sources to regulate body temperature. But I do want you to note that there are some plants that can raise temperatures in different areas of the plant, like in their flowers, and they can do that to attract pollinators. So this is something that's just strictly related to animals. It can happen in plants as well. We know that as long as there's excessive free energy, then organisms are going to be able to grow and reproduce. If there's a lack of free energy, organisms aren't going to grow and they're not going to be able to reproduce. And if there continues to be an insufficient supply of free energy, then it'll result in death. Most organisms are reproducing whenever free energy levels are at the highest. So when we look at plants, and plants have different reproductive strategies, annuals just live 1 year and they reproduce and then they die. And biennials take 2 years, 1 year to grow, and then they reproduce the second year. And perennials will continue to live year after year, but if you notice, all of them are reproducing whenever there's a lot of free energy available. So for the summer annual, it's reproducing at the end of the summer into the fall time. The winter annuals are going to grow over the winter, but they're not going to reproduce until spring or summertime. Biennials are reproducing in that summertime, and same with the perennials. You're going to see them reproduce summertime, fall time, when there's still plenty of free energy to support those structures needed to reproduce. Just something else to note in here is that it's usually not temperature that indicates whenever organisms should reproduce. It's usually the day length. So there's receptors that actually help organisms determine how long the day is, and that gives them an indication that there's going to be plenty of free energy available to reproduce. But we can have those warm days in January if plants relied on temperature to know when to reproduce. Then they could be reproducing at times when there's not a lot of free energy, and those organisms aren't going to be able to develop. We know that animals also use daylight to determine whenever to reproduce. And the fact that they can actually pause and they don't reproduce all year long is known as reproductive diapause. And again, this is happening because they're trying to time their reproduction to a time when there's plenty of free energy. So you can see that there's signaling that goes on, and it's going to determine whether organisms are going to be able to reproduce at different times or not. You don't have to have this diagram memorized, but I just wanted to make the point that plants can respond due to the length of day, and so can animals. It's an easy concept to understand that if there's insufficient free energy, it's going to result in death, but you need to be able to figure out that That's the conclusion they're wanting you to make whenever they give you problems like this and data like this. This is a question prompt and a data table that's released from 2013. And you can see that they basically eliminated the free energy supplies of a plant, a goldfish, and a sea anemone. And you can see that over time, in that day, the mass decreased. So you should understand that these organisms are actually using organic molecules to provide their free energy to maintain homeostasis. And as they use those organic molecules, they're actually going to see a loss of mass. Now, there's different strategies for organisms to obtain free energy. We're first going to take a look at producers. Producers have the ability to capture free energy and make their own organic molecules. There's two types of autotrophs. Those are chemoautotrophs. The auto part of this word simply means that they're able to fix carbon. They're getting carbon from inorganic CO2, and they're able to fix it into an organic molecule. So autotroph in plants, when we refer to plants, means the exact same thing, is they're getting carbon from an inorganic source, so that CO2 in the atmosphere, and they're converting it into organic molecules. Now, the first part of the word, photo or chemo, refers to where the energy comes from to actually allow these organisms to fix carbon into organic molecules. Photoautotrophs are using light energy to provide that energy to fix the CO2, whereas your chemoautotrophs, they're getting free energy from inorganic compounds. But both photoautotrophs and chemoautotrophs are important because they're basically the bottom of the food chain, or that food pyramid, and they are making these organic molecules that can be consumed and passed to other organisms in that food chain. Now, hopefully this diagram still looks familiar to you. When we learned about photosynthesis, we basically learned that there's two steps. There's the light-dependent reactions, and there's the Calvin cycle, or the light-independent reaction. I just want to remind you that the Calvin cycle, in this step right here, this is where CO2 is getting fixed. So CO2 is being converted into organic molecules, and it takes energy to do that. So energy comes from the ATP that's produced in the light reaction, and to convert CO2 to organic molecules, we have to reduce it. We have to add hydrogen and electrons, and those are supplied by that NADPH molecule. So autotrophs are able to fix carbon, but heterotrophs are not able to. So heterotrophs, or consumers, they're relying on getting free energy from the organisms that they eat, and they're also getting their carbon compounds as well from those organisms that they eat. So when they eat an organism, they can break down some of these organic molecules like carbohydrates and lipids. They can use it for free energy, but they're also getting their monomers, or their building blocks, to actually build their own organic molecules, their own lipids, carbs, and proteins. So hopefully this diagram still looks familiar to you. So whenever you eat food, you break it down into monomers, and those monomers can be reused by you to build your own organic molecules. Heterotrophs can further be classified as herbivores, carnivores, omnivores, scavengers, and decomposers. Of course, herbivores mean that the organism is eating plants, carnivores eat meat, omnivores will consume both. Scavengers, think of anything that will dig through your trash. That's basically a scavenger. You probably think of a raccoon, or a possum, or a mouse, or a rat, and they're different from decomposers. Now, both types of heterotrophs are consuming usually dead organisms, but decomposers are a little bit different. They usually don't have like a mouth with teeth that can tear the food. They are usually smaller organisms, or we're talking about microscopic organisms, but they're important because they recycle nutrients back into the ecosystem. When we talk about the two different types of decomposers, the detritivores and the saprotrophs, the detritivores are referring to like the smaller insects that are breaking down small organic molecules. And then when we're talking about the microorganisms that break down dead and decaying organic matter, we're talking about fungi and bacteria. Another big difference is that these are going to be internal digesters, and the fungi and the bacteria are normally external digesters. In the last part of our notes, we talked about organisms need free energy to build molecules and maintain homeostasis. And so we're going to look at how they're able to get matter so that they can build these organic molecules. So we're going to go through the different cycles: carbon cycle, nitrogen cycle, and phosphorus cycle. Now, when you see a diagram of these cycles, it doesn't mean that you need to commit it to memory, but you do need to understand what the arrows mean and be able to identify what is occurring in some of these different processes like photosynthesis, respiration, what we mean by combustion. We're going to start with CO2 in the air, and we've already talked about autotrophs are able to fix CO2 into organic compounds, and that's through the process of photosynthesis. Some of these organic compounds are actually going to get stored in the producers, and they're available to the next organism on the food chain. And some of those organic compounds, like glucose, is going to be converted into ATP, or it's going to be broken down so they can generate ATP, and that process releases the carbon from these organic molecules as the organic molecules are broken down step by step, and that carbon is released as CO2 back into the atmosphere. We know that consumers are going to consume these organic compounds containing carbon. They're going to use some of them and store some of them, then use some of them for energy. Again, that's going to release CO2 back into the atmosphere through the process of cellular respiration. This arrow just simply means that there's going to be other consumers that are going to consume organisms further up the food chain and receive those organic compounds from them. We have plant death, we have consumer death, and those organic molecules are going to be passed along to the decomposers. Just like you consuming food, they consume those organic molecules. They convert some into ATP, and that process of breaking down glucose to generate ATP is going to release carbon back into the atmosphere as CO2. If organic compounds aren't completely broken down, then they can be converted to fossil fuels. And when we burn fossil fuels, then we're going to release CO2 back in the atmosphere. Just even like driving your car, you're burning gasoline, you're going to release some CO2 into the atmosphere. After going through the carbon cycle, you should have a good idea of how to fill in these blanks. If you want to pause the video and try it on your own, feel free, and then you can check yourself. We talked about plants are fixing carbon into organic molecules. Heterotrophs are going to consume these organic molecules. Some of these organic molecules are going to get broken down to generate ATP. In the process, carbon is going to be released as CO2. Some of these organic molecules are going to be stored, and that's what we mean by assimilation. And these are the organic molecules that are available to the next step on the food chain. Whenever plants and animals die, then decomposers are going to consume them. And again, they're just doing respiration. They're generating ATP, breaking up those organic molecules, and they're going to release CO2 into the atmosphere. We can have the formation of fossil fuels if organisms aren't completely broken down. And anytime we burn any organic molecule, then we're going to release CO2 back into the atmosphere. So one common mistake that students make is that they will say that whenever organisms die, these organic molecules are returned to the soil, and then plants are able to take up organic molecules through their roots, and that's completely inaccurate. I need you to make sure you understand that all carbon to make organic molecules through photosynthesis is coming from CO2 in the atmosphere, and plants are actually taking in that gas through an opening in their leaves called a stomata, and we're going to talk more about those in probably the third segment of notes. You need to be able to look at diagrams similar to this and understand what is flowing from one organism to the next in an ecosystem. And so this is actually showing the flow of carbon through the different trophic levels, and initially this might look confusing, but the basic idea behind this is what goes in must come out. So matter cannot be created nor destroyed. So you can see here with the producers that they're passing on amounts of carbon to other trophic levels. And so if you look, they're passing on 500 grams per meter squared of carbon. So you can deduce from that is that these autotrophs over time were able to fix 500 grams per meter squared of carbon. A question that we can ask is how much carbon is released as CO2 into the atmosphere through respiration from the herbivores. If you notice, we have 125 grams going in. There's 5 grams being passed on to predators when predators are consuming some of these herbivores. Through waste and death, 60 grams of carbon is being passed to decomposers. So basically what is left, it would be 60 grams. We know that there's 60 grams per meter squared of carbon being released into the atmosphere as CO2. Now in the biochemistry unit, we talked about the 4 types of organic molecules, and they're all carbon-based because carbon is a very stable element. It can form covalent bonds, which are very strong bonds. It can bond with 4 other atoms at a time. It can bond with itself. So it's a really good backbone for all these organic molecules because you want to be made of stable molecules. So we have those 4 types of organic molecules: the carbohydrates, lipids, and proteins and nucleic acids. And I just wanted to, one, review the structures a little bit with you, since our test is only about a month away, and so that you can understand that the basic of all these molecules is carbon. So going into the nitrogen cycle, initially it looks pretty complex. Again, you don't have to recreate it, but you need to know what is going on if I say nitrogen fixation is occurring or denitrification is occurring. Again, we're going to start with atmospheric gas. We're going to start with nitrogen in the atmosphere, and our atmosphere is primarily made of nitrogen. Just like there is carbon fixation, there's nitrogen fixation, but this is done by bacteria. It's not done by plants. Plants are not able to take in nitrogen gas from the environment and convert it to a usable form. So they are reliant on bacteria to do this. We have bacteria that's fixing nitrogen from the atmosphere into usable forms like ammonium. And ammonium can be converted through a couple steps here. It can be converted to nitrate, and it can be converted to nitrate. If you ever have to memorize this, sometimes I remember that nitrate is late. So there's nitrite and there's nitrate. The one that comes second, the late one, that's the nitrate. Nitrate is late. So plants can only utilize nitrogen when it's in certain form. Plants can take up ammonium. They can also take up this nitrate. And they're doing that because they need to build these organic molecules that contain nitrogen. Amino acids and nucleic acids have nitrogen. I'll show you that here in a little bit. We also have bacteria that can take forms, molecules containing nitrogen, and convert them back into atmospheric nitrogen. And that process is called denitrification, or basically kind of undoing what they previously did whenever nitrogen fixation was occurring. Again, we talked about what assimilation means. Is plants are making these nitrogen-containing compounds. They're storing them. They're building cell structures out of them. And those nitrogen compounds are now available to any consumers that eat that material. So just like plants, animals are not able to take nitrogen out of the air and use it for any purpose. It has to come to them from their food. So whenever animals are creating waste, then those nitrogen atoms are becoming available for bacteria again to break them down into usable forms for plant. Most people don't realize that urine is actually due to the breaking up of amino acids, which I'll show you down here. So whenever you consume too many amino acids, then your body doesn't need all of them at that time, or if you are lacking glucose, your body can actually use amino acids to generate ATP. But first, this amine group has to be removed. And so this amine group is actually toxic, and it forms ammonia. And so your body has to get rid of that because it's toxic. And that's the primary component of urine, is ammonia, which is due to the breakdown of amino acids. This molecule right here that results can actually enter a step in respiration and can be broken down further so that the mitochondria can produce ATP. So you should be able to recognize some of these words, like when we say nitrogen fixation is occurring, you should be able to say that plants are taking nitrogen out of the atmosphere and fixing it into organic molecules, especially proteins and nucleic acids. And that's the form of nitrogen in the atmosphere. These nucleic acids and proteins are stored, and they're available to consumers. And like I mentioned, any unused amino acids or amino acids are being broken down to produce energy. That amine group is going to be removed, and that's going to form a waste that organisms have to get rid of. I put the 4 different organic molecules here, not because all of them need nitrogen. It's actually so you can kind of see the ones that really do need nitrogen. So we have to have nitrogen in amino acids, but we do not need nitrogen for carbohydrates. We don't find nitrogen in lipids, but we do find nitrogen when we look at the nitrogen bases in nucleic acid. All organisms need to take in some form of nitrogen so that they can make those organic molecules that contain nitrogen. So since we've been talking about bacteria fixing nitrogen and they have a relationship with plants, this is a good time to talk about that relationship. Now, anytime there's a close and permanent relationship between two organisms, that's called a symbiotic relationship. And basically what that means is, since it's a permanent relationship, where you find one, you're always going to find the other one. So when we look at plants, like legumes, like bean plants, if you pull up that plant, you're going to notice these nodules that are on the roots. And we do not see these nodules on all types of plant roots. And in those nodules, there's actually a bacteria that's called rhizobia. And that rhizobia is a nitrogen-fixing bacteria, and it's fixing nitrogen into a usable form for those plants to use. Since both organisms are benefiting, this is called mutualism. Those bacteria are getting a place to live, and the plant is getting nitrogen in a usable form. So with the phosphorus cycle, again, be able to look at a diagram, understand what is occurring, and a main idea that you need to know about phosphorus, which is a little bit different from carbon and nitrogen, is that it does not exist in the atmosphere. It doesn't exist in a gas. All phosphorus is going to be in rocks and in the soil, and it can be dissolved in water. You need to remember that phosphorus is needed for those nucleic acids. So organisms have to take in phosphorus so they can build those nucleotides. Remember, nucleotide has three parts. There's the phosphate group, there's a sugar, there's a base. And some lipids, not all lipids, need phosphorus. So that's why on the picture of the molecules here, I included the phospholipids so that you can see that there's a phosphorus atom right there. And you can see that there's no phosphorus in carbohydrates, there's no phosphorus in proteins, and there is a phosphorus in our nucleic acids. So after going through these cycles, then hopefully you can figure out the organic molecules that would go in each blank. So which organic molecule just has carbon, hydrogen, and oxygen? That's going to be your carbohydrates. Which one contains carbon, hydrogen, oxygen, and nitrogen? That would be your proteins. Which one has phosphorus, carbon, hydrogen, oxygen? Some lipids, and actually most lipids are just made of carbon, hydrogen, and oxygen. So don't always look for a phosphorus atom. And then your nucleic acids will have carbon, hydrogen, oxygen, and phosphorus, and nitrogen atoms making up their molecules. Since we're talking about getting matter from the environment, let's review real quick surface to volume ratio. We know that cells have to have a high surface to volume ratio to be efficient at taking up materials. And so this is a good place to talk about plant roots. So whenever we look at a cell that's lining the root, we're going to see these extensions of their cell membrane. And that is what we mean by root hairs. So those nitrogen atoms that are in the soil, those phosphorus atoms that are in the soil, they can be taken up by those root cells. And those root hairs give them a high surface to volume ratio to be able to adequately absorb those elements from the soil. On the third part of our notes, we're going to talk about disruptions. We're going to talk more about homeostasis. So we're going to take a second, since we've been talking about nitrogen and phosphorus, to talk about how these two elements in excess can actually disrupt an ecosystem. So if you've ever driven around and you've seen a pond and it's completely covered in algae, that's a good example of eutrophication. In that freshwater system, there is an excessive amount of nutrients, and it's usually due to fertilizers being put down on fields or on lawns, and then when it rains, the nitrogen, the phosphorus, they're going to flow into those freshwater ecosystems. And just like nitrogen and phosphorus causes plants, terrestrial plants to grow, it's going to cause algae to grow. That's why you get these ponds that are completely covered in algae, and they don't look like anything that you want to swim in. Now, just like with any population, there's a carrying capacity. So when the population of these plants get too high, then there's not enough resources to support them. And so there's going to be death and there's going to be decomposition. Now, we already talked about decomposers. When they consume organic molecules, they're going to break down those organic molecules to make ATP. So they're going to do respiration. Now, respiration requires those organic molecules, but remember, it also requires oxygen. When the decomposers are doing respiration, they'll actually remove a large portion of the oxygen from the water, and that can lead to fish kills. So that's a problem whenever there's an excess of nutrients in ponds and in lakes. So in this last part of our lecture, we're going to continue on with ecological concepts related to Big Idea 2. Remember, Big Idea 2 states that organisms need a constant supply of free energy so they can grow, they can reproduce, and they can maintain homeostasis. So we're going to look at, even whenever there's a change in the environment, how organisms are going to be able to respond so they can maintain homeostasis. So we're going to look at changes in factors that are biotic and abiotic in the environment. Remember, biotic means living, and abiotic means non-living. So the change in light would be an abiotic factor. Now, when the environment changes, organisms can respond because those changes are actually going to activate different signal transduction pathways. So remember, there's three main steps in signal transduction pathways. There's reception of the signal. That change in the environment will be detected. So the organism will receive the signal. There's going to be transduction of the signal, and then there's going to be a response. So we're going to take a look at how changing light is going to affect the growth of a plant. If you see phototropism, photo means light, tropism means growth. So if you ever notice that if you put a plant in front of a window, it looks like it bends towards the window over time. So what is actually occurring is in the stem of a plant, the side that is actually in the dark, so let's show light coming from this direction. The side of the stem that's in the dark is actually going to elongate more, and that is what causes the plant stem to bend. This response is due to the plant detecting a stimulus, which is light coming from one direction. And what happens is that a hormone called auxin, and plants make hormones just like we make hormones, auxin is actually going to migrate to this dark side of the stem. And when it does that, it's going to bind to receptor proteins on cells in that area. And it's going to activate a signal transduction pathway. Here's auxin, and it's binding to a receptor protein. So that's what we mean by reception of the signal. You can see that whenever auxin binds to this receptor protein, it's going to activate a molecule that's inside. Here's that molecule. And that molecule is commonly referred to as a secondary messenger. And remember we talked about a secondary messenger like cyclic AMP. And we said that cyclic AMP is actually made from ATP whenever two phosphates are removed. And cyclic AMP can activate other proteins. Remember, these signaling proteins are commonly called kinases, and it activates them by phosphorylating them. And they can go and they can activate other molecules as well. Now, since when activated one molecule can go and activate many molecules, then the signal grows or it's amplified. So we'll see molecules activating other molecules in the cytoplasm, and then eventually there's going to be a response. And in this diagram, the cell is able to respond in three different ways. Now, you don't have to have these memorized, but you need to have an understanding of how these events can take place, and you need to be able to understand what's going on enough that if I alter a pathway, you should be able to predict what's going to happen. But you just need to kind of understand the big ideas of signal transduction pathway and what some different responses could look like. In this first response, it says that a proton pump is activated. Here's the proton pump. It's actually moving hydrogen ions. Remember, proton means hydrogen ions. Hydrogen ions are being moved into the cell wall. Now, the reason why this is occurring is because this cell is going to have to elongate and most likely divide. Because again, we're trying to get more growth on one side of the stem so that the stem is going to bend towards the light. Whenever we're moving hydrogen ions, we're actually making that cell wall more acidic, and it's going to break up. Now, the cell wall is made of cellulose, and so since we're going to need more of a cell wall as the cell elongates and divides into two, then the Golgi can be activated by the secondary messenger, or it can be activated by a kinase. It just depends on the pathway. And you can see that the Golgi is responding, that it's releasing these vesicles that contain the substance that the cell membrane is made out of. You should be able to recognize what's going on in this part of this diagram. It's showing you that this secondary messenger has bound to another molecule that's symbolized in red. And this would be a good example of a transcription factor. So signaling molecules can bind to transcription factors. And so then you can see transcription is taking place because the mRNA is getting made. And so then the mRNA moves out to the cytoplasm after the mRNA is processed. Remember, it gets a cap and a tail, and the introns are spliced out. And so then translation will happen, and these different proteins are made, and these proteins are related to elongation and division of the cell. Again, so one side of the stem can elongate. So let's fill in on number 3. These secondary messengers or these kinases can activate a transcription factor, which will lead to the production of proteins. We've talked about before the different types of chemical messengers or hormones or signaling molecules or ligands. To review very quickly, remember that your lipid hormones, your steroids, since they are lipid soluble and the cell membrane is made of lipids, like dissolves like, so lipid hormones can diffuse right through the cell membrane, but note, they're going to bind to a receptor, but that receptor is in the cytoplasm. Whereas your protein-based hormones, they're charged and they're large. They're not going to make it in, so they're going to have to bind to a receptor protein on the surface of the cell, and you can see that happening right here. This is a water-soluble or a protein-based hormone. It can't make it through the membrane, so it's going to bind to a receptor protein that is embedded in the plasma membrane. In this unit, we're going to do a transpiration lab. And again, we're focusing on ways that organisms are able to maintain homeostasis. And so we know that it's really important that organisms can regulate their water levels. So we're going to take a look at how plants can regulate their water levels. Just like you have blood vessels that transports blood, a plant has transport vessels to transport water and sugar. So the xylem is the name of those transport vessels that transport water. The phloem transports sugar, but we're not going to focus on that, so you don't have to worry about that. So water actually moves from roots to leaves and not vice versa. I have some students that believe that water is absorbed through leaves, and that does not occur. So water moves from the roots to the leaves, and it does so due to 2 main forces. The first one is called cohesion tension. And we're going to review water properties a little bit here. So if you remember, water is a polar molecule. So those hydrogen and those oxygen atoms are sharing electrons because they're covalently bonded, but they're doing so unevenly. So we get these partial charges in different areas of the molecules. So whenever you have other water molecules with these charges, these opposite charges attract. And if you remember, that is why hydrogen bonding occurs between these molecules. And the fact that 2 water molecules are attracted to one another, that's called cohesion. So what happens at the leaf of a plant is there are openings that are called stomata. So the stomata are these openings in the leaves, and you're going to see a picture of them here in just a little bit. But they're openings in the leaves so that the plant can exchange gases with the atmosphere. Plants need to bring in CO2 to do photosynthesis, and they need to get rid of excess oxygen. But gases can move through these stomata, and so can water. So plants can lose water through the stomata. And when this happens, whenever water evaporates through the stomata, we call this process transpiration. You need to memorize what transpiration means. So what happens is, at this leaf, at the stomata, when one water molecule leaves, it actually pulls that next water molecule up in its place. And this water molecule is going to pull the next water molecule up into its place. And so you get this tension that pulls the whole column of water up in the plant. Now, this is also aided by the fact that water shows adhesion. And remember, adhesion means that water is attracted to other molecules. So gravity is trying to pull water down in a plant, but whenever water moves up in the plant due to cohesion, those water molecules are attracted to the side of the xylem. So again, it's just kind of another property that's, or another factor that's aiding in water being able to move up and stay up and not have gravity pull it back down to the roots. There's another factor involved, which is called root pressure. And we talked about that plants take in different solutes at their roots. So they're taking in molecules that contain nitrogen and phosphorus. And whenever these solutes move into those root cells, water is going to follow. Because remember, water moves to where there's a high solute concentration. So water is going to move into the roots from the soil because there's a higher solute concentration in those root cells. Now this is a good time to review water potential. We know that water moves from areas of high water potential to areas of low water potential. So now that we're talking about the movement of water through plants, you should be able to take a look at these different values for water potential and predict which way water is going to move. So soil has, according to this diagram, has a water potential of -0.3. So in order for water to actually leave the soil and go into those root cells, that root cell has to have a lower water potential. So let's put its water potential at -0.4. So water is going to continue to move from cell to cell because one cell is going to have higher water potential, a neighboring cell is going to have lower water potential, and that's why water is going to move from one to the next. If you notice when you get over here to the xylem, so this is what I mean by the vessels that water is transported in from the roots up to the leaves. The xylem has even lower water potential than those cells that are in the tissue at the root. When you get up here to the leaf, so this is kind of showing you up here, we've got a leaf here. The leaf has even lower water potential, and then when you look at the outside air, outside of the stomata, so here's actually kind of a picture of what the stomata looks like. This lower water potential causes water to move out of the leaf through the stomata and into the atmosphere. If you remember that there's two factors in water potential. There's pressure and there's solute. And to review, remember, if you increase pressure potential, then you're going to increase water potential. So think of a syringe, and if you push in that stopper and you increase the pressure in that syringe, the water is going to shoot out. So if you increase pressure, you're actually increasing the potential of water to leave. If you increase solutes, you're going to drop water potential. Remember, if you had a beaker of pure water, the water potential was 0. But if you added solutes to that beaker, then it made the water potential more negative. So since we're talking about plants regulating water, we need to talk a little bit more about the stomata since there's a significant amount of water loss through the stomates. So when you look at plants that live in different areas that have different amounts of rainfall, you're going to see that they're going to differ in the density and the location of their stomata. And that is, again, it's all due just to water availability in their habitat. So when you see areas that have plentiful rainfall, then those plants are going to have a lot of stomata. They're going to lose a lot of water, but that's not going to be a factor because it rains often. So they're not going to be deficient in water. So they can afford lots of openings in their leaves. If you have plants that are in drier areas, you're going to see more stomata on the bottom of the leaf, and there will be few, or there might even be none on top. And so when the stomata are on the bottom of the leaf, it kind of offers some shade, and you're going to have less transpiration. When you have plants in very dry areas, they'll be less dense than a plant that's in an area that has more adequate rainfall. And so if they do have those stomata, they're all going to be on the bottom of a leaf where it's shaded. If you have an underwater plant, you're not going to find any stomata. And we're actually going to do a stomate peel on a few plants so that you can see and calculate the density of stomata. So again, we're looking at how organisms can maintain homeostasis even if there's a change in the environment. We're going to see how CO2 levels, light, lack of water, and temperature, how that can actually affect the stomate and a plant's ability to regulate their water levels. So this is what a stomate looks like. And on the edge of the opening, there's actually two cells, and they're called guard cells. And these guard cells are going to control whether this hole is open to the atmosphere or it's closed off. And they do that because water is going to move in and out of these guard cells, and it's going to affect the turgidity of these guard cells. Remember, turgidity is just another way to say turgor pressure, and we're talking about pressure on a cell wall due to water. And so whenever a plant detects that it has low levels of CO2, or if there is a receptor that is detecting that light is present in the environment, then the stomata is going to open. A plant needs to exchange gases with the environment so that it can do photosynthesis. So you can kind of hopefully understand why it's an advantage when a plant detects light and knows to open those stomata, because it can now do photosynthesis. So it needs to take in as much CO2 as possible so it can make those organic compounds. So what happens is that light is going to activate this proton pump that is in the cell membrane of these guard cells. So there's the proton pump right there. And what happens is this proton pump pumps hydrogen ions out of the guard cells. And what this is going to do, this is going to set up an electrochemical gradient. So basically what that means is you're going to have an area that has a positive charge more so than another area, and that other area is going to have more of a negative charge. These potassium ions in this situation are going to move down the electrochemical gradient. They're positively charged, so they're actually going to move to the area that is more negative. So all these little red dots are trying to symbolize potassium. So you can see there's a bunch of potassium inside of those guard cells. Whenever you get a high solute concentration in the cell, water is going to move into that cell. With all the potassium ions inside of the cell, water actually moves into the guard cells, and those guard cells are going to swell. They're going to get real tight, and then you're going to see that opening there, the stomata. What's happening? Let's go ahead and fill in some stuff in our notes here. The light activates a proton pump. Remember, proton is just a fancy way to say hydrogen ion. Those hydrogen ions move out of the guard cell, setting up that electrochemical gradient. This is going to cause those potassium ions to move down the electrochemical gradient, and that high solute concentration is going to cause water to move into those guard cells. Again, this is a process that it's not so important that you memorize all of these steps, but everything that I said should make sense to you. So if you read a different scenario, then you should be able to figure out what's going to occur. The more examples I give you, the more likely you're going to be able to answer questions related to scenarios. The stomata can also be closed, and so this is going to happen whenever a plant becomes stressed, and a plant usually becomes stressed because it's lacking water. It releases a hormone, because remember, plants can release hormones as well, called abscisic acid. It also closes stomata whenever it detects really high temperatures in the environment. If the plant is deficient in water, the stomata is going to close simply because there's not a lot of water available to fill up those guard cells. So when there's not water in the guard cells, those guard cells are real spongy, or they're real flaccid. And so they actually move together, and they're kind of closing that opening there. So that lack of water is just going to lead to low turgor pressure. So this is a good example of negative feedback, when the end result is actually going to stop this process. So for example, the plants open their stomata, and this is the end result. It ended up in water loss due to transpiration. So this water loss, in turn, can actually turn off the opening of the stomata, and the stomata are going to close. Now, when we think of transpiration, we probably think of it as being a negative thing, and it is, because most plants don't have a plentiful supply of water, so they have to closely regulate the amount of water that they're losing. But there is a benefit, because whenever water evaporates, so let's write evaporative cooling, it actually cools off the plant, just like whenever water evaporates from your skin, it cools you off as well. In this lab that we're going to do, you're going to be given a plant and you're going to alter its surroundings. So some things that we're going to do is we're going to put some plants in the dark, we're going to put them in front of a light, we're going to put them in front of a fan, we're going to try to heat them up by putting them in front of a heater. We can even try to put a plant in natural light versus artificial light, and we're going to see how it affects transpiration rate, the amount of water that is lost over a period of time. The method that we're going to use is easy and it's simple, and it's not going to look quite like this. This is sometimes difficult to set up. You have to have an airtight seal, and if you don't, water leaks out and you're not going to get good results. But if you see this setup, then you should know that the people that are conducting this lab are trying to measure transpiration, or the amount of water that's lost by a plant over time. So what happens is you take a cutting of a plant and you put it in this tube, and this is filled with water. So you can see the water level right here. And as the plant is taking in water and water is moving up due to cohesion tension, then that water is going to leave through transpiration. And so as water leaves, more water is going to move into the plant, and this level is going to drop. So you can actually tell how much water a plant is losing due to transpiration over time. So let's talk about some things that should increase transpiration. Well, you probably figured out that if you put a plant in a windy environment, it's actually going to increase the amount of water that it's going to lose. It's going to dry out that plant. Wind should increase transpiration rate. We also are going to put plants in light and dark areas. And so if you remember, we said that light actually causes stomates to open, and when they're open, that's whenever plants can lose water. So light should actually increase transpiration rate. One group is going to be given a bag, and they're going to place it over their plants, and we're going to spray water into that bag so that we can create an environment that has high humidity. And so if the environment is really humid, then the plant is going to lose less water. So we're going to see decreased transpiration rates in high humidity. Now, we talked about if we increase temperature, a plant's going to close its stomata because it'll lose a lot of water in high temperatures. So we should see that if we increase temperature, we're actually going to see a higher rate of transpiration. Even though those plants are going to close their stomata, they're still going to lose a lot of water just due to high temperatures. And if we have light increasing transpiration, then the plants that we put in the dark here, we should see a decreased rate of transpiration.